Hello, young generation. I hope everybody is fine and going strong with your health and mind. So as you can see, today we are going to view the second video on the same chapter, Microbial World, Friend and Foe. So we are going to study these three topics for the day. Number one is what are carriers and vectors. Then what do you mean by food preservation? And of course, the most important topic among the whole is nitrogen cycle. So today we will cover up these three topics in this video. So please sit with your books and notes copies and other materials so you can write down the notes as the tutorial proceed. Now first of all, let us see what do you mean by the term when I use the word what are carriers, okay? So since we know we have got lot of pathogens or the microbes that affect our health, so this topic will let you know how these organisms enter our body system. So first, what is a carrier? So underline and write down the answer. So these are those organisms which carry the infectious agents and they serve as a potential source of infection, okay? So they are the organisms that harbors or in which the infectious agents live, okay? And they act as a potential source of infection. So that is as far as carriers are concerned, okay? Then what are then vectors? Okay, these again refer to an organism, okay, that spread disease from the pathogens into the host body, okay? So they carry these pathogen or the disease causing microbes from one body to another without causing disease by itself. So they won't cause disease by itself because they only act as a carrier, or you can say a mediator between the two different hosts. So examples are lot many. We will deal with them as we move into the topic of these carrier and vectors as the tutorial proceed. Now before we move into that, we should know what are the similarities and dissimilarities between them or differences between them. So as far as similarities is concerned, so you can see the vectors and carriers are the two types of agents. So they are the agents that transmit disease between the organism, and both these vectors and carriers are involved in the biological mechanism of transfer of diseases, and that means that they again transmit the disease causing microbes. So what we call the disease causing microbes as, so we call them pathogens, okay? So both of them transfer the pathogens and cause diseases. So in this case again, disease causing microbes may live on inner body of both vector and carrier. So these pathogens can live in the body of both carriers as well as vectors. So that is as far as their similarities is concerned. Now let us see what is the main point. So that has to be written by you. Key difference between carriers and vectors, okay? So write down them after pausing the video once the tutorial is over. So in case of carriers, is an infected organism capable of transmitting diseases to other organisms, okay? Rest of the part is not meant for you. Just to write out, carrier is an infected organism, so which is capable of transmitting the disease, okay? While what are vector? So they are the organism which transport the disease from from the infected person to healthy person, okay? So they act as a intermediating agent, so which transfer. So the carrier has a disease while vector itself is disease free, okay? So they only act as a medium of transportation. For example, for carrier is HIV, that you people know causes AIDS, though we will study about them in detail in coming years to come. While in case of vectors, we have the example in your textbook regarding the mosquito, that is female Anopheles mosquito. Female Anopheles. So we will again read this word, but for the timing you write it down. Female Anopheles mosquito, okay. It causes the disease called malaria. So these two terms should be known to you, that what are the similarities between the two, vectors and carrier, and what are the differences between the two, okay. Now let us proceed further. So as I have told you, in case of vector, we have this female Anopheles mosquito, write it down. Female Anopheles mosquito, which carries a parasite called malarial parasite. And write down the name of that malarial parasite, that is called Plasmodium. Write it down, Plasmodium, Plasmodium vivax. Sometimes we get this question here, 1 mark question. And after writing the answer, underline these names separately, okay. Female Anopheles mosquito carrying the parasite of malaria called Plasmodium vivax, which goes into our bloodstream and causes the disease called malaria. So that is for vector, okay. And in case of carrier, so there is a virus called HIV. You can write it down. Human, full form for HIV, human write it down, immuno deficiency virus, okay. Immunodeficiency virus, okay. And that causes the disease that is being told to us is AIDS. So there is a lot of controversy about it, but since you are small children, you just write it down that the disease caused by HIV is AIDS, okay. That is the example for carrier. Now comes the next topic called food preservation, okay. So write it down that what is food preservation. So the food preservation is the process of treating and handling food with an aim to stop or slow down its spoilage while maintaining its natural value, texture, flavor, that is the taste, okay. Since the microorganisms causes the deterioration of food, they also call something, as you will read in your textbook, called food poisoning. So as a result, we have to food poisoning. So as a result, we have to develop some mechanism so that the food get preserved. So that is what is the food preservation. So it is the step or the process so that we can slow down, okay, the spoilage while maintaining the nutritive value of food, its texture and flavor. So these are the main agenda for food preservation, and for that we use many methods. So we will take the methods one by one as the tutorial proceed. So as I have told you that food preservation, we prevent or remove microbial contamination, or we want to retard the microbial growth so that we will have minimized food spoilage, food infection and food poisoning. So I have just told you this data, so that just underline again and write them again. So because we need food preservation to minimize food spoilage, infection and food poisoning. So there are a number of methods to prevent food from the spoilage or the contamination. First, we all consume it, so you should be knowing it. The first method is called pickling, okay. And the food preserved in this process is by a chemical called vinegar. So as a result, we can also use the same chemical to preserve meat or vegetables due to action of, underline the word, acetic acid, which prevent the microbial growth, okay. So vinegar has the acetic acid in it, and that prevent the microbial growth and the food get preserved. Then we have one more method that we are using for ages and ages called salting. So the food is preserved by it because the salt prevents the microbial growth, because it dehydrates, it takes the water content out of the food stuff. So DEHYDRATE, dehydrate. So when there is no water, the microbial infection get drastically reduced. Then we all have this apparatus at home, you can see it. So we call the process as refrigeration. Why? Because at low temperature again, the microbial infection get limitized and get approximately nullified, you can call it. So we also refrigerate our food articles. Then we have this methodology that should be learned by you in detail called pasteurization. And this pasteurization is a technique developed by Louis Pasteur, hence the name pasteurization. So in this what we do is that we heat the food stuff, generally the milk, and chill the heated milk immediately. So as this temperature fluctuates, the pathogenic bacterium that is in the food or in milk get eliminated. So basically in this process we use the heat to kill the microbes. So this process again generally you should be knowing from your textbook. We will study about that process, that we will heat the milk at about 72 degrees C, okay, for 15 seconds, and then immediately cool it down. So at this temperature fluctuates, the microbial flora in the milk can't sustain it and they get themselves eliminated. Then we have many other methods under the heading called chemical additives. So in case of chemical additives, we use it for preventing the food against spoilage and to add flavor to enhance the taste, okay. So it serves two purposes. It prevents the food from spoilage or contamination and also add flavor to it. Now, there are various methods that we have just done for preventing the food from spoilage, which I have told you. Dehydration, take the water out, underline the word. Definition: dehydration means removal of water from the microbial aspect, and as the water is removed, the moisture is removed, so they can be prevented or they can be preserved for a longer span of time. So basically we use this process for pulses and cereals, okay. So they act as a big methodology for preventing against contamination. Then I have already told you refrigeration and freezing. So because these microbes like bacteria and fungi cannot live or thrive at low temperature, so basically we use this technique for fruits and vegetables, okay. So refrigeration and freezing generally we use for that because the microbes can't tolerate that lower temperature. Then we have other techniques for storing the food. One is called canning. So in canning again we take airtight cans that affect as a good process for preserving them. So basically jams and vegetable and fishes are preserved by this technique. Then we have this methodology that we have in the book also, chemical preservatives, okay. So these chemical preservatives are again the substances which preserve the food and hence called preservatives because they control the microbial growth of the various microbes that contaminate our food. And so we have some names in that you have to learn them. So one is sodium benzoate, okay. So write down this name. That name is still in your book. Sodium benzoate, okay. So write down the name properly, and again you can check the book. Sodium benzoate and sodium metabisulfite, okay. Sodium metabisulfite, sodium meta. So these names are new for you people, so please learn them properly. Sulfite, okay, PHITE, sodium metabisulfite or potassium metabisulfite. So these are some names that you should be knowing it. So number one was sodium benzoate and sodium metabisulfite or potassium metabisulfite. So these are the chemicals we use for food preservation. Then in this, as I have told you, we have some salts. So one is benzoic acid, those who want to study better. So as I just told you, sodium benzoate is the one that we use to preserve the food article like fruit juices, jams and jellies, because they act as good preservative. Then in same chemical preservatives, we have this age-old process. We use sugar and salt, which I have just told you. So that thing should be known to you, the various salts and the sugars. And then also you can have some name that is been in your respective textbook also, lactic acid, okay? Lactic acid. So they all act as food preservatives. Then sometimes we do ask you name some gaseous chemicals we use for food preservation. So write down these two names, sulfur dioxide and carbon dioxide, okay? Sulfur dioxide is SO2. This is the formula you might have read last year. And carbon dioxide, CO2. So they are also acting as a chemical preservatives. Then I have just told you that sugar and salts are also used because these chemicals inhibit the organism itself, the growth of there. As a result, they draw the water from the tissues of the food. As a result, when they get dehydrated, the water is taken out, the food is preserved for a longer span of time. Now come this particular diagram that you have to draw from your book, and since the diagram is a bit more tough for your age students, so this is the simplest of the diagram that you can check it out. So here the nitrogen from the atmosphere is fixed by a bacterium called Rhizobium. You can see that name in your book also. So there are lot many fixation that will take one by one. Then it goes into the soil, then plant absorb it, and when the plant dies off, the nitrogen goes back to the nature. Plants are also consumed by animals, and when animals die, again it goes back to nature. So make sure whenever you get something to draw in cycle form, it should start and end at the same junction to know how the cycle proceed. For people, those who are more in the scoring zone, because this same topic will study in 9th standard also, for them I will prefer they should understand and draw this diagram, okay? So just check out the data, that will give you more understanding that how this process proceed. So again, number one is nitrogen in atmosphere, okay? So this nitrogen comes into the living form or into the system or cycle through three processes. One is by nitrogen fixing bacterium, the name we have just told you, Rhizobium. Number two is atmospheric fixation during lightning, and this is industrial fixation, okay, when we use it to make fertilizers. So in all these three processes, nitrogen, biological fixation, we call it biological fixation through Rhizobium. Then when lightning strikes, so we have atmospheric fixation and industrial fixation. Ultimately, this component comes into the system of cycle. So in nitrogen fixing bacteria, they convert it into the respective zone of ammonia, and in case of atmospheric fixation and fertilizers, they come in the form of nitrates in the soil, okay? Nitrates, underline this word, we are going to use it too much for this class. Nitrates and come into the soil, and these nitrates are then used up by the bacterium under the process called denitrification, denitrification. So DE nitri, NITRIFICATION, okay, denitrification. And after this denitrification process, they get back into the atmospheric nitrogen format. While some of the nitrates are absorbed by plant, they take it as to make the food, and we have a word here called metabolism. So there are new words coming for you. metabolism, okay, metabolism, that is something concerned with reactions in the body. And when they take it for metabolism, then plants are also consumed by we people as a food, so come into animal metabolism format. And when living organism remove their waste, they again turn into ammonia. And then is a new word called nitrifying. Okay, earlier it was denitrifying, here it is nitrifying. Okay, F-Y-I-N-G, nitrifying bacterium that put it back into the soil. So we have lot many words in this, so we'll take them one by one as we proceed in the tutorial, but I will prefer that you should learn this diagram and pause the video, see the data and draw the whole diagram, because we'll again going to study the same cycle next year. Now let us see what the theory is. So in case of nitrogen fixation, as I have told you, it takes place through three processes. One is atmospheric fixation, where the lightning breaks nitrogen into nitrogen oxides, okay, atmospheric fixation, that is. The number two is industrial fixation, where we combine nitrogen with hydrogen to form a compound called ammonia, okay, form the compound called ammonia. And this ammonia is then utilized for the making of lot many fertilizers such as urea. So basically this is the process under the heading industrial fixation. And then comes the biological fixation, in which I have told you the bacterium called rhizobium, and the bacteria like rhizobium and the blue-green algae, okay, they convert the nitrogen into usable form for living organisms through various terms and definitions. So underline these three processes by which nitrogen is fixed, okay. Number one was atmospheric fixation, industrial fixation and biological fixations. So these are the terms we will be repeating too much in this year and next year also. Now underline how this process of nitrogen cycle get fixed, though we have just told you. Now you should know these terms, meaning of that. First is fixation, that is the first step, making nitrogen usable by plants. Then I have just told you in diagram nitrification. So write down this word again, nitrification, in which process by which ammonia gets changed into nitrates. The new word is called nitrates. We have just wrote that word in that tutorial earlier. So nitrates, that is called nitrification, conversion of ammonia into nitrates, okay, ammonia into nitrates called nitrification. And then the third process is called assimilation, that how the plant get nitrogen. And the last process here is called ammonification, that is the decaying process, when the living things die, so their body get decomposed and turn into ammonia. So these are some steps for nitrogen fixation. Now since that process is bit more complex and lengthy, so you cut short and write down the things as shown in the board, okay. So as I have told you in nitrogen fixation, underline some data and write down the data. The first step is the nitrogen cycle. So this step is conversion of atmospheric nitrogen into ammonia, okay. N2 is nitrogen, NH3 is ammonia, and who will do it for us? Again, the beautiful microbe called rhizobium. Then we have lot many processes I have just told you. First one was assimilation. Underline the definition, the nitrogen is fixed in the soil so that plant can absorb it, and we call that process as assimilation, okay, assimilation, ASSIMILATION, okay. The second process is called ammonification. So we have just told you. So the process ammonia is generated and then is taken up into the soil, by the plants to grow. So it is just a release or conversion of animal and plants when they die off into ammonia, referred as ammonification. Then we have called the process denitrification and nitrification. So in case of nitrification, as the process explain you the answer, you should know that in nitrification what happens, I just told you. In this, basically the ammonia, NH3, okay. So ammonia is converted into, first is nitrite, write down the new word, nitrite, Nitrite, and from nitrite it is converted into nitrate, okay. From nitrite it get converted into nitrate. So the terms looks like similar, but they are quite different. So in this process, the ammonia get converted into nitrite first and then into nitrate. So that is a process called nitrification. And for denitrification, what happens? So in this, the nitrate which is being absorbed by the plants is converted into nitrogen back into the gaseous form like NO2, that is nitrogen dioxide, okay. So denitrification means conversion of nitrates back into nitrogen oxide or compounds of nitrogen called NO2, nitrogen dioxide. So as you can see, the whole process, the nature takes its own turn and take all the components that you have taken from the nature take its back. So it is called nitrogen cycle. Now the question arises that why we should have the nitrogen cycle? What is the importance of it? So write it down properly. So in this particular format, as we have these cycles, so we can have, because it brings the inert nitrogen from air into biochemical process in plants. So it convert the inert, means non-reactive nitrogen into the form which the animal kingdom or plant kingdom can use. Then number 2, so plant need nitrogen to make, underline the word, chlorophyll, because with chlorophyll they can do the process called photosynthesis. So you see it is a very important process. So with nitrogen the plant make this chemical called chlorophyll. And during the process of ammonification, I have told you, the body get decomposed and naturally it act as a cleaning up of environment. So you can write this point in Microbial World also. So the microbe help to clean the environment by this process. Then we have some other applications due to the nitrogen cycle. You can see the nitrates, I used the word and nitrites earlier, are released into the soil, which help the nutrient pool of the soil. So soil get more nutrient from it. And write down these two words, nitrates and nitrites. And then plant use this nitrogen compound for their integral part of cell composition. And due to this again, so we can make our own cell living, because you know nitrogen is a big component for making our protein molecule also. So by looking into the whole data, again refer the book first and then go through this tutorial, draw the diagrams and make sure to learn them also. As a result, it will help you out in the whole process of discussion and its helping hand. So take care, bye.